Hi friends, you're back with me, Professor Girish Kukreja, your friend for microbiology, biotechnology, biochemistry. I hope you remember me, yes, because it's been too long that we have met here. But yes, I'm back here. Today we'll be talking about this test here. One word which I can see here everywhere is the Ames test, right? So here I'm not be describing the test in detail, but some of the questions, like when you're performing the Ames test, then what are the questions which may be asked during your viva or during your interviews. We'll discuss one or two questions here. And yes, but if you think that I should describe the entire test, please mention in the comment box, right? So as I told, not the entire test, but yes, but you'll get a glimpse of the test from here. Okay, without wasting much time. The test organism used in the Ames test, right? We know that the Ames test is used to detect the mutagenic ability of a particular compound, because we know that mutation is linked to cancer. Oh, so the test organism, yes. So without wasting much time, the most probable answer, no, the only answer here you can say is the bacteria Salmonella typhimurium, right? So the test organism which we use is a auxotroph. Remember, auxotroph, an organism which requires a particular growth factor in the medium for its growth. It doesn't have the ability to synthesize that growth factor. Yes. So this is a histidine auxotroph. So the answer would be a histidine auxotroph of Salmonella typhimurium. Now there are different strains, like you have TA1537, TA1538, which has a frame shift mutation in the histidine synthesis genes, or you have TA1531, which has a mutation, which is a point mutation for histidine synthesis. Anyways, it should not be able to synthesize histidine. So that is the test organism used here. Okay. So how are the conditions in the mammals mimicked while performing the Ames test? Yes. So we know that when a particular compound is being tested, this does not actually represent the compound as it is, because many compounds, when they enter our body, they could be metabolized and then they may be turned carcinogenic, or vice versa, that if it is not carcinogenic, after metabolism it may turn carcinogenic. Bruce Ames in 1970s, he took proper care of this, and they came up with the idea that we can use a rat liver extract. So the conditions are mimicked by using the rat liver extract. It is generally the S9 fraction which is used to mimic the conditions. You take the compound, mix it with this rat liver extract of S9 fraction especially, which will cause some metabolism, and then you could actually mimic about what is happening to these particular compounds inside the human body. This is one of the limitations actually, but then yes, we will talk about it when we are describing the entire test. Next one, state the basic principles of the Ames test. The basic principle behind the Ames test is that your organism, your Salmonella typhimurium, right, which is histidine negative, if after addition of your compound gets converted to an organism which is histidine positive, that means it can grow in a medium without histidine. It has undergone some mutation and has reverted back to its prototrophic strain. So, if a particular compound can cause a mutation reverting back from S typhimurium histidine minus to S typhimurium histidine plus is a potential mutagenic agent and hence a potent carcinogen. So that is the basic principle behind Ames test, where we check that whether this histidine auxotroph is reverted back to its wild type prototrophic variant, that is histidine positive. So if it is a mutagen, it is probably a carcinogen, right? So that is the basic principle behind the Ames test. Comment on the medium used in the Ames test. Yes, obviously we are trying to search for an organism which is histidine positive. So we'll prepare a media which does not have histidine. So if it is not having histidine, obviously the colonies, if I get on this plate, right, then they are prototrophic, or they are now not auxotrophic for histidine, because they are growing. It means that they are synthesizing histidine. It means that mutation has taken place in that gene for histidine synthesis, from where it has grown from histidine minus to histidine plus. But one characteristic feature of this medium, you have to remember that it contains limited or a very less minuscule amount of histidine, because for mutation to take place, for mutation to proliferate, And to get it shown, you need the organism to multiply so that the DNA gets replicated and it gets some ample time for expression of this mutation. So the medium, it contains very less amount of histidine, which allows a minuscule growth of the organism so that the mutation continues, and then you can find these colonies. So the characteristic feature of the medium is that it contains a very less amount of histidine. Okay, so stay tuned with me, Professor Girish Kukreja, with more in microbiology, biochemistry, and biotechnology. As I told, coming soon with this entire procedure of the Ames test. Thank you so much.